How much would you be willing to pay a security firm to protect you 24 hours a day for all 365 days of the year? Would that service be worth hundreds, thousands, or maybe tens of thousands of dollars? What if I told you that you already have this service and it costs you nothing? This incredible service is provided by your immune system, which is the system that recognizes and destroys foreign invaders. When your immune system responds to the presence of foreign invaders, we call it an immune response. Your immune system is a valuable asset and selflessly protects you from many different foreign invaders, which we call antigens. In this lesson, you'll learn how your immune system keeps you safe and learn about two arms of immunity, known as antibody-mediated immunity and cell-mediated immunity. Immunology, which is the study of the immune system, is not a new science. Even ancient Greek scientists noticed that once someone got sick, that person was unlikely to come down with the same sickness a second time. However, their understanding was limited by their lack of scientific equipment. In fact, it wasn't until scientists started to examine blood under microscopes that the basis of immunity was established. Scientists noticed that the blood of a person who survived an infection changed. The surviving person's blood now contained proteins in the blood that protected against future attacks. These proteins, that were produced in response to the infection-causing antigen, were named antibodies. When you have antibodies present in your body fluids that provide immunity, the immunity is called antibody-mediated immunity. This is also referred to as humoral immunity, because the medieval term for body fluids is humor, even though there's not much funny about blood, lymph, or any other body fluid for that matter. The word mediate means to intervene to try to make something better. This might help you remember what antibody-mediated immunity is, because we see that antibodies intervene and try to make your health better by getting rid of antigens. When your doctor gives you a vaccination, his hope is that the shot will stimulate the production of antibodies inside of you, resulting in this type of immunity. Antibody-mediated immunity is just one of the two arms of your immune response. The other arm is called cell-mediated immunity, which is when special cells provide immunity by ridding the body of unwanted cells, like infected cells, cancer cells, or transplanted cells. With this type of immunity, we see cells called macrophages engulf antigens. The term macrophages is easy to recall if you break the word down. The prefix macro means big, and the suffix phages means eaters. So macrophages are literally big eaters, and they have an insatiable appetite for antigens. After the macrophage eats the antigen, it chews it up and presents the antigen fragments on its surface. Specialized white blood cells called helper T cells encounter these macrophages and send out signals that call different cells, like cytotoxic or killer T cells, into battle. These killer T cells rapidly divide and develop into an army of cells ready to fight and destroy the unwanted invading cell that triggered the immune response. Let's review. Your immune system is a system that recognizes and destroys foreign invaders, or antigens. An immune response is what happens when your immune system responds to the presence of foreign invaders. Antibodies are proteins in the blood that protect against future attacks from antigens. When you have antibodies present in your body fluids that provide immunity, we call it antibody-mediated immunity. When special cells provide immunity by ridding the body of unwanted cells, we call it cell-mediated immunity. This type of immunity involves special cells like macrophages, helper T cells, and killer T cells.
Take just a minute to glance at your surroundings. Do you see them? They are everywhere around us, and there are more of them than there are of us. Who am I referring to? Well, it's not who, it's what. I'm talking about microorganisms, also called microbes. They are the organisms that are too small to be seen with the naked eye. This means that you don't see microorganisms unless you're looking at your surroundings through a microscope. This is the instrument used for viewing things that are too small to be seen with the naked eye. Now that you know they are there, you need to know what to do about them. The good thing about microorganisms is that not all of them are bad or harmful to us. The microorganisms that are not harmful are referred to as non-pathogenic. Non-pathogenic microorganisms are useful to us in many ways. Some help us create foods we enjoy like cheese and bread, while others help us digest food. Since they are helpful, we aren't very concerned with them. We are concerned, however, with the microorganisms that are harmful and capable of causing a disease process in the body. We refer to these as being pathogenic. Pathogenic microorganisms are the ones that can infect or cause disease in or on our bodies. These are the ones that we need to wage an infection control war against in order to stop their ability to infect the body. As a nurse, you are responsible for your part in the infection control war to protect yourself and your patients. We establish now that we want to stop pathogenic microorganisms from infecting our bodies and the bodies of patients. One key thing to doing this is knowing what it is microorganisms want and need. Our infection control strategy is to cut off what they need to survive. The first thing they need is the right temperature. Every type of microorganism has a set temperature at which they thrive. Some prefer warmer, while others prefer cooler temperatures. The vast majority of microorganisms grow and thrive best between 25 and 40 degrees Celsius. We have to keep that in mind because the human body maintains a temperature of 37 degrees, which is perfect for most pathogenic microbes. The next requirement is pH. This refers to the acidity or alkalinity. The preference for most pathogenic microbes is close to the neutral range of 7.0 to 7.4. Our bodies are set to a pH around 7.4. Another requirement is moisture. The available amount of water determines whether microbes can make and transport food as well as get rid of waste. They always require an ample supply of water. We are 70 to 80% water. Speaking of food, that brings us to their next requirement. Microorganisms must have nutrients. This gives them what they need to make food and get energy. They are able to extract the nutrients they need from the foods we digest. The fifth thing that all microbes need is the right mixture of gases. This is the air composition in the area where they are growing. The different gases that may be needed or restricted include oxygen, nitrogen, carbon, and hydrogen. Guess what? We have areas in our bodies that have all combinations of these gases. The last thing they need is the right amount of light. There are pathogenic microbes that thrive in the dark as well as those that thrive in the light. The surfaces of our body and even certain areas inside contain the right amount of light for them to grow. Remember that you are one of the first lines of defense in infection control as a nurse. You know what the pathogenic microbes need, so you need to stop them from getting it. The number one way to stop an infection from occurring or spreading is to always wash your hands using antimicrobial soap between patients and anytime you enter or leave a medical facility. This includes going to the restroom and leaving to eat. This takes away their nutrient supply and changes the pH. There are a number of other ways to fight the battle against pathogenic microorganisms. These are provided for you in the CDC's list of standard precautions. Be sure to become familiar with each precaution so you know how to stop infections from spreading. The main thing is to keep them from getting into your or anyone else's body. Now that you are winning the war, let's look at the microbes you are fighting against. Bacteria are the one-celled microbes with few internal structures. Some examples of pathogenic bacteria are Staphylococcus aureus, which can cause toxic shock syndrome and cellulitis, Escherichia coli, which can cause hemorrhagic colitis, and Borrelia burgdorferi, which causes Lyme's disease. Viruses are another type of microbe that you've defeated. These are molecules of DNA or RNA inside of protein that require a host to live in. Almost all viruses are pathogenic. Some familiar viruses include human papillomavirus, which can cause genital warts and cervical cancer, Norwalk virus, which can cause gastroenteritis, and herpes zoster, which causes shingles. Fungi are another type of microbe. These are single or multi-celled organisms that are categorized as yeast and molds. Common fungi include trichophyton metagophytes that cause tinea commonly called athlete's foot, and Candida albicans, which causes oral thrush. The next defeated microbes are the protozoans. These are a group of one-cellular organisms that behave like animals. The most notorious of protozoans are the Plasmodium species. There are about 5 or more of them that cause malaria. We covered some very important responsibilities that you're going to have to take on as a nurse. Some basic terms that we discussed include microorganisms, also called microbes, organisms that are too small to be seen with the naked eye. Microscope, instrument used for viewing things that are too small to be seen with the naked eye. Non-pathogenic, microorganisms that are not harmful. Pathogenic, microorganisms that are harmful and capable of causing a disease process. We then went on to discover the 6 requirements for microorganism growth. The 6 are temperature, pH, moisture, nutrients, gases, and light. We ended the lesson with looking at the 4 main types of microorganisms. The 4 are bacteria, one-celled microbes with few internal structures; viruses, molecules of DNA or RNA inside of protein that require a host to live in; fungi, single or multi-celled organisms that are categorized as yeast and molds; protozoans, one-cellular organisms that behave like animals.
We previously learned that the body works hard to regulate and control blood flow. But if a blood vessel gets injured, the body must jump into action to avoid the loss of too much blood. In this lesson, you will learn about the events that happen immediately after a wound to control bleeding. Almost immediately after you suffer a cut, your body reacts by initiating a series of events that happen one after another until the bleeding stops. Hemostasis is a term used to describe the stoppage of bleeding. Now, this is an easy term to recall if you remember that heme is the Greek word for blood, and stasis is the Greek word for halt. So hemostasis can be literally thought of as the halting of blood or the stoppage of bleeding. This series of events depends on specific proteins called clotting factors. Clotting factors are substances in your blood that act in sequence to stop bleeding by forming a clot. Now, there are many clotting factors, and they often interact with each other. Many of them are named using Roman numerals, such as factor 12, factor 11, factor 10, and so on, while others go by more common names such as fibrinogen, or prothrombin, or tissue factor. And we'll learn more about these clotting factors later in the lesson. Now, these clotting factors depend on vitamin K to function properly. Vitamin K is required for the synthesis of clotting factors, and it's so important to the blood clotting process that it sometimes goes by the nickname the clotting vitamin. Without it, you could experience excessive bleeding, and so vitamin K is important to get in your diet, and it's found in such foods as green leafy vegetables and broccoli, cauliflower, cabbage, so there's another good reason for you to eat your vegetables. Now, when a blood vessel wall is damaged, collagen fibers from within the wall are exposed, and these exposed fibers become a place for platelets to cling to. Now, platelets are irregular shaped bodies that help the clotting process by sticking to the lining of the blood vessels. Now, these odd-shaped fragments of cells are normally found floating around your blood along with your red blood cells, kind of minding their own business. But when the cells that line the blood vessels get injured, they release chemicals that cause the platelets to kick into action and become sticky. And what we see is layer upon layer of platelets laying down over the wound, somewhat like dirty plates piling up in a sink. Platelets are like the first responder to a site of an emergency, but platelets can't plug the hole alone. They need some help. This is where we start to see some of the clotting factors that we talked about earlier. And one in particular, called fibrinogen, is an inactive clotting factor that helps bind the platelets to form a clot. These inactive clotting factors act as little crosslinks, attaching the adjacent platelets to each other. So in wound healing, platelets take a lot of the glory for being the first ones to the scene, but without their support team, fibrinogen, they would not be able to properly hold together. So we're starting to see how a wound can get plugged up with platelets. But the thing is, there's not enough platelets in the immediate area of the wound to properly plug the hole. So platelets solve this problem by calling for help. So how do platelets call for help? They do this by releasing tiny granules that send out chemical signals. And these chemicals attract more platelets to the wound site, which helps build the plug. But these chemicals do more than just recruit more platelets. For instance, one important chemical in this process is called thromboxane A2. Thromboxane A2 is a product of activated platelets that promotes the collection of more platelets and vasoconstriction. Vasoconstriction is an important component of wound healing, and it's one of the very first steps in healing a damaged blood vessel. It's this vasoconstriction that initially slows the flow of blood through the vessels and decreases the amount of blood lost. So thromboxane A2 is a platelet recruiter and a vasoconstrictor, making it an important player in blood clotting. Interestingly, though, aspirin is an antithrombotic agent. And we know that anti means against and thrombo means blood clot, so aspirin is a drug that is against the formation of a blood clot because it blocks the synthesis of thromboxane A2. Without the thromboxane A2, the platelets can't keep coming to the injured site as quickly, so aspirin can lead to more or prolonged bleeding. So we already see that wound healing is a series of events that picks up momentum as it goes. The activated platelets start plugging the wound and send out chemicals to attract more platelets. We previously learned that this is known as a positive feedback loop because it is the type of feedback that results in an increase or amplification of the effect by its own influence on the process. In other words, we can say that the process of healing promotes more healing, and this positive feedback accelerates blood clotting and speeds the overall healing process. So at this point, the wound has a temporary plug to stem the bleeding, but it needs something more permanent. Tissue factor, or factor 3, is a clotting factor that is activated due to the damage of a blood vessel wall. This starts a cascade of events that causes one clotting factor after another to activate, somewhat like a waterfall. And this waterfall of events is known as the coagulation cascade, and is defined as a series of reactions involving clotting factors that stop bleeding by forming a clot. Now, if you've ever stood beside a powerful waterfall, you realize that the water is going to fall no matter what you do. The same thing goes for the coagulation cascade. The clotting factors continue to activate each other one after another until they reach the bottom of the waterfall. And while it's not important to learn each of these clotting factors, we can focus on the clotting factors at the bottom. And here we notice that we have the inactive clotting factor prothrombin that changes to its active form thrombin. Thrombin then in turn catalyzes the conversion of fibrinogen to fibrin. Fibrin is at the bottom of our coagulation cascade, and indeed it can be thought of as the last step because it is fibrin that's the biological glue that seals the damaged vessel and ensures hemostasis. It does this by forming long strands of fibrin threads that crisscross heavily and form a fibrin mesh over the original wound site. Let's review. Hemostasis is the term used to describe the stoppage of bleeding. Hemostasis is really a series of events that depends on the specific proteins called clotting factors, which are substances in the blood that act in sequence to stop bleeding by forming a clot. Vitamin K is required for the synthesis of clotting factors. When a blood vessel wall is damaged, one of the first things to happen is vasoconstriction. This slows blood flow to the damaged area. The damaged blood vessel also releases chemicals that cause platelets to become sticky. Platelets are defined as irregular-shaped bodies that help the clotting process by sticking to the lining of the blood vessels. These platelets get a little help to hold themselves together from fibrinogen, which helps bind the platelets to form a clot. Activated platelets also send out for help by releasing chemicals to pull more platelets into the area. Because of this, we can say that blood clotting is accelerated by a positive feedback loop. Thromboxane A2 is one of these chemicals released by the activated platelets that promotes the collection of more platelets and vasoconstriction. Aspirin can block thromboxane A2 and may lead to increased bleeding. Now that the wound is temporarily sealed, the body can start work on a more permanent healing through the coagulation cascade. This is a series of reactions involving clotting factors. The cascade continues until we see the activation of fibrin. It is fibrin that is the biological glue that seals the damaged vessel and ensures hemostasis. It does this by forming long fibrin threads that crisscross heavily and form a fibrin mesh over the original wound site.
Whether it's due to sports or a medical problem, or maybe an accident like a fall down some steps, you might get a nasty wound. A wound is just an injury to an organ or tissue of the body that results in a disruption of the normal continuity of that organ or tissue. That's a mouthful, but I know you can relate to this because you've certainly had a wound on your skin before. The skin is an organ of your body. The wound, let's just say it was a cut, disrupted the normal cohesion or unity of the skin. The skin was broken, and that's what I mean by a disruption of normal continuity. That wasn't hard to understand, was it? Probably not. In that case, let's move on to the 3 phases of wound healing. I promise I'll make it easy on you. The first phase of wound healing is the coagulation or inflammatory phase, or just both coagulation and inflammatory phase. Sometimes this phase is actually split into 2 separate phases. However, it's easier to combine it into one because both of these aspects of this phase start to occur at pretty much the same time. Coagulation is another word for clotting, and inflammation is a local protective response to an injury that helps to dilute, destroy, or wall off the injured tissue or the agent responsible for the injury. Important signs of inflammation include pain, redness, heat, swelling, and in some instances a loss of function of the area in question. What happens in this phase is as follows. After you cut your skin, the broken blood vessels of the skin begin to gush out blood. Not good. The body does not want to lose blood. That's why platelets, cells that plug up holes in blood vessels, stick to the damaged blood vessels and initiate a cascade of steps that results in the clotting of blood. The clotting of blood plugs up the holes in the broken blood vessels and the bleeding stops. Yay! The platelets, as well as other cells, also release chemicals that help attract inflammatory cells called white blood cells into the area. These guys are here to ensure that whatever agent, be it chemical or biological, which has caused the injury or entered into the body as a result of the injury, is destroyed or walled off behind a jail-like compound. The white blood cells also release inflammatory compounds that cause you to experience the signs of inflammation I just went over. As the inflammation eventually subsides, the second phase of wound healing, the proliferative phase, begins. The inflammatory cells that I mentioned before release compounds called growth factors. These growth factors are ultimately responsible for the formation of granulation tissue. This is a fancy term for tissue formed to repair soft tissue wounds. You can think of it as a natural patch or band-aid your body tries to create to protect and repair the wound. Something else happens during this phase. Remember how I said the first phase can be split into 2 phases because of overlapping events? Well, pretty much all of these phases of wound healing overlap in some way or another. It's one continuous and overlapping process. One key example of this is that of angiogenesis, the creation of new blood vessels, which begins in phase 1, but doesn't really take off until phase 2 in importance. That's why I stuck it in this section. Angiogenesis is super important. If a bunch of underwater pipes that water the grass on top of them burst, the grass will die without new pipes being installed, right? Well, the injury that took place burst and incapacitated a bunch of your body's internal pipes, the blood vessels. If the injured area is to avoid dying off like the grass, the body grows new pipes, new blood vessels, to support wound healing. Why are these blood vessels important? It's because they provide nutrients for cells to grow and repair the wound. Once a normal blood supply has been reestablished, the third phase of wound healing, the maturation or remodeling phase, can begin. Here, the wound is constantly altered in how it looks, the cells that make it up, and even its strength. This is the part of wound healing where you see scars appear. What's important to remember about this phase is that remodeling proceeds slowly. This means that the strength of the newly deposited tissue increases slowly over a pretty long period of time, months to a couple of years. In most cases, the wound does not recover the full strength of the original tissue after the remodeling phase is complete. Most wounds are about 20% weaker than the original tissue. However, some tissues like bones or the bladder may regain 100% of their original strength. Let's wind this wound lesson up. A wound is just an injury to an organ or tissue of the body that results in a disruption of the normal continuity of that organ or tissue. Once you've been wounded, the first phase of wound healing, the coagulation/inflammatory phase, can begin. Coagulation is another word for clotting, and inflammation is a local protective response to an injury that helps to dilute, destroy, or wall off the injured tissue or the agent responsible for the injury. In this phase, bleeding stops and inflammation kills or tries to kill any harmful agent that may have entered your body. The second phase of wound healing, the proliferative phase, begins thereafter with the formation of granulation tissue, protective tissue that helps repair and protect the wound. This phase is also the one where angiogenesis, the creation of new blood vessels, is super important to support the granulation tissue. Finally, the third phase of wound healing, the maturation or remodeling phase, occurs thereafter as the wound changes slowly over time and scar tissue may develop.
At some point in your life, you've probably been stung by a bee, got hit in the arm, or scratched yourself. In any of these three scenarios, you likely experienced pain, swelling, redness, and other things in response to a damaging stimulus, be it the bee's stinger or your friend's knuckles. All of these bodily reactions to the harmful stimulus are classified under a collective term known as inflammation. Yet the big question is always, why does inflammation occur and how does it start? How does our body actually recognize this damaging stimulus and induce a rapid response to it? That's what this lesson will answer. Anything that acutely, that is to say rapidly, damages your body in any way, internally or externally, can induce inflammatory responses, including redness, pain, swelling, heat, and even loss of tissue or organ function. These damaging effects can be due to a wide variety of things. Pathogens such as bacteria, fungi, viruses, and parasites, molecules such as toxins and drugs, foreign proteins in the form of antibodies, and physical trauma. In one way or another, any and all of these can alone, or in some cases together, trigger the inflammatory response within your body. The list of triggers can impact your body in a wide variety of ways. Let's go over one very important way. Let's say someone punches your shoulder or cuts it, or bacteria invade it and release toxins that begin to destroy its tissues. Regardless of the cause, the result is damage, physical or chemical, to the cells of your shoulder's tissues, such as those of the skin or muscle. Each cell in your body contains a protective envelope called a cell membrane. In this cell membrane, there are important constituents, one of which is called arachidonic acid. Arachidonic acid is a fatty acid found in cell membranes that is released from cell membranes during normal metabolic processes, cell death, or injury. To illustrate its release from the cell membrane, let's go out to your garage, pick up a sledgehammer, and beat your car door silly with it. That impact of the sledgehammer and resulting physical trauma will cause bits and pieces of metal and paint and dirt to be released into the air and onto the ground below. That's what happens in cellular damage as well, regardless of the initial cause. Once arachidonic acid is released from the cell membrane, it plays a critical role in inflammation. Derivatives of arachidonic acid that are critical to modulating inflammation are formed. These derivatives are collectively called eicosanoids. There are two major forms of eicosanoids I want you to be aware of in general. I know all of these names sound weird, but it's important for later lessons when we learn why they play a key role in many different treatments for many disease states. Leukotrienes are one type of eicosanoid. Leukotrienes are eicosanoid inflammatory modulators formed thanks to the enzyme lipooxygenase that play many roles in inflammation. As I just said, leukotrienes have many roles in inflammation, such as the dilation of local blood vessels. Another role is the recruitment of leukocytes or white blood cells such as neutrophils to areas that need them. Hopefully you can remember this easily since the prefix leuko, meaning white, is present in both leukocytes and leukotrienes. Use this trick to remember at least part of what leukotrienes do. Basically, the leukotrienes serve as a source of bait for white blood cells to come to a local area of damage. There, the white blood cells can attack foreign bacteria that may be dangerous to the body. They'll also secrete all sorts of compounds, such as histamine, that will dilate local blood vessels, which will cause further redness and swelling in the area and allow for even more immune system cells to pour into the area to fight off the foreign invader. As you can tell, the roles leukotrienes play may overlap with many different substances in the body. This redundancy ensures a strong immune response to a traumatic event, but also plays a role in disease states related to an abnormal immune system, such as allergic reactions. In addition to leukotrienes, prostaglandins are produced as well. These eicosanoid inflammatory modulators are formed thanks to the enzyme cyclooxygenase and play many important roles in inflammation. These roles include the mediation of pain, dilation of blood vessels, and the increase of white blood cells in circulation, among others. While you may think I've already overloaded your brains with too much information, we haven't even scratched the surface of the roles arachidonic acid and eicosanoids play in inflammation, let alone other cellular processes, nor the true complexities of how they interact with one another. What's more, we haven't even touched upon compounds such as acute phase proteins, nitric oxide, complement proteins, interferons, interleukins, chemokines, and beyond. All of which play important roles in inflammation and all of which can be triggered in very similar ways to what I described in this lesson. The end point here is something, anything, can cause your body's cells to sense that they are damaged or their neighbors are damaged, and this triggers a cascade of events that results in the immediate effects we feel and see, those which we term acute inflammation. All sorts of lipids, amino acids, carbohydrates, and glycoproteins exist in your body that either initiate, prolong, or stop the process of inflammation. What's also important to note is that most of these are synthesized and activated at the local site of injury and are quickly and locally inactivated, all in order to try and minimize the sometimes terrible repercussions of a body-wide or systemic inflammatory reaction, which we'll cover in another lesson. It's basically like this. You may be wandering in the woods and all of a sudden feel really cold. Being really cold is very bad for your body. So you start a little fire next to yourself in order to make yourself feel better. However, you quickly put it out right there and then as soon as you feel normal. If you started the fire all over the forest right away, or you didn't put it out properly, the whole forest would burn down. Therefore, your body acts in a similar way. It tries to only start a fire that it needs to, for only as long as it needs to, in order to avoid damaging your entire body. That's because inflammation is indiscriminate, and just like fire, it hurts you as much as it hurts whatever it is that is causing your cells injury if it gets out of control. So, let's just remember this. Something causes an injury to a cell in your body. This cell will release arachidonic acid. Arachidonic acid is a fatty acid found in cell membranes that is released from cell membranes during normal metabolic processes, cell death, or injury. Arachidonic acid is then used to form eicosanoids, which are derivatives of arachidonic acid critical to modulating inflammation. These eicosanoids include leukotrienes, which are eicosanoid inflammatory modulators formed thanks to the enzyme lipooxygenase, and prostaglandins, which are eicosanoid inflammatory modulators formed by the enzyme cyclooxygenase. Both eicosanoids play many roles in inflammation. Our body has many ways it protects itself. Your skin is a big barrier to the dangers of the outside world. Stomach acid can kill most pathogens. Your immune system is there to fight off everything from bacteria to viruses. This lesson explores the flip side of the latter points. That is to say, your immune system, via the inflammatory process, may end up hurting you in the long run. Under normal circumstances, if something harmful causes inflammation in your body, that inflammation will dissipate relatively quickly after the stimulus for its initiation disappears. That's exactly how it's supposed to occur. But sometimes chronic inflammation occurs. Chronic refers to diseases that continue for a long time or recur over and over again. This chronic inflammation can be very harmful to your body. In fact, I know you're not surprised by it. Imagine dropping a 10-pound weight on your toe over and over again, or wrapping a rubber band over your finger and never taking it off. Those types of continuous events would harm your body quite a bit. In the same manner, chronic inflammation harms your body in a wide variety of ways. Inflammation refers to the sensory consequences of inflammatory mediator release. These mediators include protein, carbohydrate, and lipid compounds and derivatives which initiate, promote, prolong, or stop the process of inflammation. These mediators can either directly damage your body or indirectly damage your body by recruiting other mediators or cells to the local site of injury. For instance, white blood cells that are recruited by inflammatory mediators to a site of injury may release degradative enzymes that end up destroying not only foreign bacteria, but your own body cells as well. Therefore, inflammation can physically or chemically hurt you. Furthermore, inflammatory mediators also cause you pain via their stimulation of nerve cells. Anyone with chronic back pain knows exactly what I'm talking about. The causes for chronic inflammation are as many as the biochemical mediators that initiate and prolong this process. Here's one particular illustration of this. Usually, when a virus enters your body, it will trigger the inflammatory response. This inflammatory response seeks to destroy the virus. However, in select cases, some viruses can enter inside of your body's cells and essentially hide there, thumbing their noses at your immune system, right under the watchful eyes of the inflammatory mediators and cells. This happens for several different reasons, but the end result is a process of chronic inflammation that seeks to destroy the virus that's hiding inside the cell. While the inflammation is triggered for a good reason, to destroy the virus, it ends up hurting the body far more than the virus itself. Since the virus sitting inside the cell can't be killed very well, the inflammation continues indefinitely, giving rise to everything from pain to organ failure to cancer to death. The reason cancer can be a result of chronic inflammation is because inflammation relies heavily on something known as reactive oxygen species, which may be secreted by inflammatory cells such as neutrophils. These reactive oxygen species are molecules and free radicals derived from oxygen that are involved in the process of inflammation. The reactive oxygen species may be really good for things like killing bacteria, but they can also be terrible in the long run because they can cause damage to a cell's DNA. If a cell's DNA is damaged and very specific genes are damaged enough, then the cell may become a cancer cell, divide, and then spread around the body, killing the person. Another way inflammation can become chronic and hurt a person is in the case of autoimmune diseases. Autoimmune diseases are conditions where the body's own immune system causes it to attack itself. Sometimes there is a genetic cause for this, but other times it may be that the body's immune system is tricked into thinking that its own cells are the bad guys. So, as soon as the immune system is fooled, it launches a terrible inflammatory attack on all sorts of tissues and organs throughout the body, many times resulting in incapacitation and eventual death of the person. Now you hopefully have a better appreciation for how inflammation can hurt you instead of help you when it comes down to chronic inflammation. Chronic refers to diseases that continue for a long time or recur over and over. Chronic inflammation occurs thanks to prolonged inflammatory mediator release. These mediators include protein, carbohydrate, and lipid compounds and derivatives that initiate, promote, prolong, or stop the process of inflammation. Some of these mediators play a role in the release of reactive oxygen species, which may be secreted by inflammatory cells such as neutrophils. These reactive oxygen species are molecules and free radicals derived from oxygen that are involved in the process of inflammation. In another lesson, I mentioned the importance of realizing that most inflammatory mediators are made and inactivated at the local site of injury in order to try and minimize deleterious body-wide effects of inflammation. While this is true, it's often harder to achieve the latter point than not, since your body isn't an isolated series of compartments. Everything interacts everywhere all of the time, and sometimes this is actually necessary. A case in point is the flu. It's a respiratory infection, but the inflammation induced by the flu virus makes your entire body feel terrible. However, that terrible feeling may actually be important for your recovery, as you'll soon learn. In this lesson, we'll discuss the body-wide or systemic effects of inflammation, why they occur, and what may happen in extreme cases. One very well-known component of systemic inflammation is known as pyrexia, or fever. The way I recall this definition is by thinking about the fact that a pyromaniac is someone who loves to set things on fire. The first three letters of each word match. Fire is hot, and I know you felt very hot when you've had a bad fever. As a general rule, you must recall that fever is usually associated with an infection, but you can impress your teacher by telling them that people can have a fever due to many other things, such as cancer. Anyway, there are two important reasons for the fever. A high temperature is great for killing off pathogens, and a high temperature speeds up important protective chemical reactions. Therefore, while the fever may make you feel bad when you've got the flu, you should actually thank it effusively for helping you kill off whatever it is that's trying to hurt you. However, you surely know that a fever can be a bad thing, since a prolonged high temperature can cause a person to feel miserable, may cause seizures, and in serious cases, even death. What's also critical for you to know is that the major internal initial trigger of fever in our body is a protein known as IL-1, or interleukin-1. It is mainly secreted by white blood cells called macrophages in response to a foreign pathogen. This trigger of fever, or pyrogen, IL-1, then causes the synthesis and release of a compound known as prostaglandin E. This is a lipid compound that ultimately tells the hypothalamus to raise our body's temperature. The hypothalamus is the structure in our brain that regulates our body's temperature. As you can tell, there's a whole sequence of events that leads to a fever. The IL-1 is like a match that is sparked. This spark then lights up the tissues, prostaglandin E. The little flames of prostaglandin E then trigger the firewood, our hypothalamus, to light up and produce powerful heat all over the place. Besides causing fever, another major systemic response to inflammation is known as leukocytosis. This is an abnormally high white blood cell count in the blood. This occurs as a result of compounds called epinephrine and corticosteroids. In short, they cause an increase in the white blood cell count by forcing mature white blood cells, called neutrophils, to enter the circulation from storage pools inside of the body. By forcing the neutrophils to get out there and get going, so to speak, they have a much higher chance of encountering whatever it is that is causing you inflammation, destroying it, and getting you back to normal ASAP. Basically, epinephrine and corticosteroids act as a swift kick in the butt to a lazy teenage son, the neutrophils, who need to stop lying around on the couch and should go outside and save the world like Batman. Now, while I did mention two important systemic effects of inflammation, I don't want to leave this lesson alone without mentioning the fact that systemic inflammation can also be a bad thing if it's not kept in check. One of these bad effects is known as cachexia. Cachexia is the wasting away of body fat and muscle as a result of chronic disease processes or inflammation. One reason cachexia occurs in chronic disease states is because an inflammatory mediator known as tumor necrosis factor alpha is released for too long a time. One thing this inflammatory compound does is that it basically causes your body to improperly utilize and absorb fat from your diet. Meaning, there may be enough food to go around, but the body just can't use it properly due to chronic inflammation, and the person ends up losing a lot of weight. Another terrible consequence of too many inflammatory mediators being released at once is known as anaphylactic shock. This is a systemic inflammatory reaction to an allergen that causes the blood pressure to drop to dangerous levels in conjunction with problematic breathing, itching, and vomiting, among other signs and symptoms. One major component of anaphylactic shock is a compound called histamine that is released by mast cells and basophils during a reaction, directly causing many of the problems I just mentioned. While mast cells and basophils are important components in inflammation, their overstimulation or overreaction may lead to death as an anaphylactic shock due to a sudden, massive, and systemic inflammatory response to something your body perceives as a threat, whether real or not. Sometimes this reaction can occur because of an innocent substance that wouldn't cause the body nearly as much harm as anaphylactic shock does. I liken this reaction to a person who worries way too much about nothing important. It's like that mother who hands you rubber boots, an umbrella, and a waterproof coat when there's only one tiny cloud in the sky. Could it theoretically rain because of that one cloud? Yes. Will it really cause that big of a problem? No. Well, now I hope you can appreciate how important and how dangerous systemic inflammatory responses can be. Two important components of a solid inflammatory response we went over were leukocytosis, which is an abnormally high white blood cell count in the blood, and pyrexia, or fever. The major internal initial trigger of fever, a pyrogen in our body, is a protein known as IL-1, or interleukin-1. This pyrogen, IL-1, then causes the synthesis and release of a compound known as prostaglandin E. This is a lipid compound that ultimately tells the hypothalamus to raise our body's temperature. The hypothalamus is the structure in our brain that regulates our body's temperature. While fever is usually a good thing, unless it gets out of hand, and leukocytosis usually indicates a normal response to a foreign invader in your body that needs to be killed, the systemic inflammatory response can go haywire. Two notable examples of this are cachexia, which is the wasting away of body fat and muscle as a result of a chronic disease process or inflammation, and anaphylactic shock, which is a systemic inflammatory reaction to an allergen that causes the blood pressure to drop to dangerous levels in conjunction with problematic breathing, itching, and vomiting, among other signs and symptoms. If you live in a big city and want to try to avoid traffic jams associated with such a densely populated place, then one of your options is to ride a subway or train to get to your destination. Each stop along the way gets you closer to where you need to go. In this lesson, we're going to take a metaphorical ride on a train that will represent our body, on a track called the disease process, from our starting point called infection to our destination stop that's called recovery. As we go on this ride, we'll discover the different generalized steps associated with disease progression. At the starting point of our train ride, called infection, we get a few nasty passengers aboard our train, our body. A bunch of microbes board the train, take a seat, and begin to multiply. The train they board is our body, and the seat they take is like a cell in our body that they use for replication purposes. Once the microbes board the train and infect our body, something known as the incubation period begins. This is the period between infection and the first signs and symptoms associated with a disease. During the incubation period, these microbes multiply all they want, and the person may actually spread the disease-causing microorganisms without even knowing they are sick. Again, this is a symptomless stage where microbes not only multiply inside of our body, but can also spread to other people. The length of the incubation period is highly variable and is dependent not only on the microorganism that invades the person, but the person's immune system as well. Once the train leaves the infection station and proceeds through the incubation period, it reaches the next stop or stage of disease development, known as the prodromal period. This is a short stage of disease development where a person begins to feel that they are getting sick. Basically, this is where our train's conductor, our brain, knows that something is wrong, but is not exactly sure what. Essentially, the body feels something is amiss, but the full onset of signs and symptoms associated with the disease have yet to fully hit us. As I mentioned, the prodromal period is quite short, and pretty soon progresses to our next stop, called the period of illness. This is the step in the development of a disease where a person feels the typical signs and symptoms associated with the disease. In short, this is when you are sick. That's basically what it means. Our train essentially breaks down at this stop. This is also the stage where you can most easily transmit a communicable disease to another person. You can imagine that once we reach this stop, a lot of the microbes that infected us a couple of stops back leave the train to seek out new targets. The very peak of an illness's intensity, where you can most easily spread around these microbes, is known as the acme point. Once we pass the acme point, we proceed into the period of decline, which is the stage of disease development where the immune system begins to bring microbial replication under control, which leads to the lessening of clinical signs and symptoms associated with the disease. Essentially, you can liken this phase to the train's engineers, our immune system, finally getting the upper hand in fighting off the disease-causing pathogens and fixing our body up enough to get us moving and feeling a bit better. Once the train gets moving a bit better, we reach the last stop of our train ride. This is known as the period of convalescence. This is the fifth and final stage of the disease process, one where microbial replication is fully stopped and the person returns to the pre-illness state. In short, this is the stage where the person recovers from their disease. However, that definition comes with a catch or two. In some cases, depending on the disease, the person may not proceed to the period of convalescence and may die from their disease or be disabled. In addition, during any period of disease, from the first stop until the very last one where you are no longer experiencing symptoms and signs associated with the disease, you can transmit the disease-causing pathogen to another person. In essence, even if you don't feel sick, that doesn't mean you can't transmit the microbe that got you sick in the first place. Before I end this lesson, I thought I'd share something cool with you. If you've ever felt like you are getting sick, meaning you were in the prodromal stage, but didn't get truly ill in the end, then you may have already been infected with whatever caused you those feelings before. In short, if you were infected by something like a virus that led you down the 5 stages of disease development we went over, your immune system would have remembered this virus for the future. This memory allows your immune system to respond to an invading pathogen far more quickly the next time around, killing it off before it causes the period of illness. That's why you felt like you were getting sick, but never got truly sick in the end. You can thank your immune system's memory for that. It's like our train's engineers remembered what caused the train to break down last time around and knew what to do and how to fix it this time around before it caused any serious problems. With that in mind, let's go over all of our train ride's stops one last time. After a microbe, such as a virus or bacterium, enters our body without being stopped by our immune system, the incubation period begins. This is the period between infection and the first signs and symptoms associated with the disease. We then enter the prodromal period. This is a short stage of disease development where a person begins to feel that they are getting sick. As I mentioned, the prodromal period is quite short and pretty soon progresses to our next stop, called the period of illness. This is the step in the development of a disease where a person feels the typical signs and symptoms associated with a disease. In short, this is when you are sick. That's basically what it means. The very peak of an illness's intensity, where you just feel miserable when you are sick and when you can most easily spread around these microbes, is known as the acme point. Once we pass the acme point, we proceed into the period of decline, which is the stage of disease development where the immune system begins to bring microbial replication under control, which leads to the lessening of clinical signs and symptoms associated with the disease. After the period of decline, we enter into the period of convalescence. This is the 5th and final stage of the disease process, one where microbial replication is fully stopped and the person returns to the pre-illness state. In short, this is the stage where the person recovers from their disease unless they were killed or disabled by it prior to fully recovering from it. If you're a fan of ancient books or of Hollywood tales, then you've undoubtedly heard of something known as the Trojan horse. This was a device used by the Greeks to outwit the Trojans during the Trojan War. In a similar fashion, we'll discover in this lesson that any pathogen or agent of disease that is out to get you uses a sequence of events similar to the one the Greeks devised to destroy the city of Troy, except these pathogens are out to destroy your body instead of a city. When a pathogen enters its host and multiplies inside of it, we term that process an infection. A pathogen can be anything you can imagine, such as a virus, bacterium, fungus, parasite, or even a prion, which is an abnormally shaped protein that causes disease. The entire process of infection can be likened to the men inside of the Trojan horse entering the city of Troy. When the horse enters the city, soldiers jump out of it or multiply out of it and infect the city. The ease by which an infectious agent can enter, survive, and multiply within a host is known as infectivity. In our case, because the soldiers inside the Trojan horse were carried into the city and easily survived and multiplied within it, they would be deemed highly infective. However, the fun has only just begun at that point. After the soldiers hidden away in the horse have multiplied out of it, so to speak, they run towards the city gates and open them up, thereby allowing the entire Greek army to come into the city. Once the soldiers of the Greek army run into the city, they begin to slaughter everyone and anyone that stands in their way. The people the Greeks kill are the citizens of Troy. The citizens of the host city are like our own bodies, cells, tissues, and organs. They are being attacked and destroyed by the pathogen. When your own body's receptors, cells, tissues, or organs are damaged, destroyed, or inactivated, resulting in an abnormal state of function, health, and overt clinical signs and symptoms, we term that process a disease. There's only one catch here. Disease is a two-way street. The citizens of Troy don't just lay down their arms as the Greeks attack. They fight as hard as they can for their home, their city, and life. Likewise, our body has something known as an immune system, which fights off pathogens trying to freely multiply within our body. The problem is, the immune system is sometimes not very picky in how it wages war. You can liken our immune system, especially in the early stages of a disease, to the citizens of Troy showering fire arrows down upon the Greeks. While the fire arrows will certainly kill plenty of enemy soldiers, those same arrows can inadvertently strike a citizen of Troy or cause a building to catch fire. Basically, they cause destruction and mayhem on both sides of the war. Likewise, inside of our own body, the immune system not only damages the invading pathogen, but also our own body cells, tissues, and organs. Therefore, our immune system contributes to the entire process of disease to a great extent. It's definitely not a perfect system by any means, as I am sure you can now clearly tell. Now, before we move on to something else, I want you to look over the definition of disease one more time. Disease is when your own body's receptors, cells, tissues, or organs are damaged, destroyed, or inactivated, resulting in an abnormal state of function, health, and overt clinical signs and symptoms. There's something missing in that definition from the context of this lesson. Nowhere did I say that a disease must be caused by an infection. I know for a fact you can think of at least one disease that doesn't have to be caused by a pathogen. The list is actually quite long, but here are some examples: diabetes, Cushing's disease, heart disease, cancer, and so on. Again, an infection is just one potential cause of disease, but not the only one. In fact, sometimes we don't even know the root cause of a disease or the etiology of it. Many times, a disease is idiopathic, meaning it's a disease that has no known cause. Finally, before ending this lesson, like the Greeks ended the Trojan War, I'd like to cover one more important point. Earlier on, I mentioned the fact that the Greek soldiers hiding inside of the horse opened the gates to the city to let more soldiers in. I did this for a reason I'll get to in just a second. You need to realize that virtually any infection that causes a disease, and even one that doesn't cause any overt signs of a disease, may lead to something known as an opportunistic infection. An opportunistic infection is an infection that results from a pathogen that doesn't cause disease in a normal, healthy host, but one that instead takes advantage of a weakened immune system in order to do so. Basically, these pathogens are little wimps that wouldn't dare take on a healthy person's immune system and instead wait for them to become immunocompromised or incapable of forming a normal immune response. Viruses are a prime example of pathogens that weaken the immune system only to allow a secondary opportunistic infection to occur. One famous disease known as AIDS, which occurs due to a virus known as HIV, is a prime example of this fact. In the context of our Trojan War, you can liken an opportunistic infection to a smaller and weaker enemy of Troy, waiting for the Greek army to swing the gates open and start overwhelming the city. Once these weaker enemies see this occurring, once they can tell that the immune system is weakened, only then do they walk through the gates to start exacting their own revenge on Troy. I know there was a lot to cover in this lesson. However, I hope you at least found the examples interesting to follow. Let's summarize all of the important parts one more time. When a pathogen enters its host and multiplies inside of it, we term that process an infection. A pathogen can be anything you can imagine, such as a virus, bacterium, fungus, parasite, or even a prion, which is an abnormally shaped protein that causes disease. Regardless of whether it's a prion, bacteria, or otherwise, the ease by which an infectious agent can enter, survive, and multiply within a host is known as infectivity. Sometimes we know that a disease is caused by some kind of infectious agent, such as the ones I listed before. Other times, we don't know the root cause of a disease or the etiology of it. Many times, a disease can even be idiopathic, meaning it's a disease that has no known cause. Finally, you need to realize that virtually any infection that causes a disease, and even one that doesn't cause any overt signs of a disease, may lead to something known as an opportunistic infection. An opportunistic infection is an infection that results from a pathogen that doesn't cause disease in a normally healthy host, but one that instead takes advantage of a weakened immune system in order to do so. Basically, these opportunistic pathogens are little wimps that wouldn't dare take on a healthy person's immune system and instead wait for them to become immunocompromised or incapable of forming a normal immune response.
How do viruses pass from one person to the next? When one child has a cold at school, why is the rest of the class likely to come down with one the next week? Let's explore how infection gets from one place to another, or the chain of infection. The chain of infection, if we think of it as an actual chain, is made up of 6 different links: pathogen, reservoir, portal of exit, means of transmission, portal of entry, and the new host. Each link has a unique role in the chain, and each can be interrupted or broken through various means. The first link is the pathogen itself. This is the disease-causing organism. For many illnesses and diseases, this is a virus or bacterium. In order to break this link, various methods can be used, including the pasteurization of milk, the chlorination of drinking water, or the use of disinfectants. The second link is the reservoir. This is the natural environment that the pathogen requires for survival. Reservoirs can be a person, an animal, or an environmental component, such as soil or water. This link can be broken through medical treatment and testing, insect and rodent eradication, or quarantine. The third link is the portal of exit. This link is needed for the pathogen to leave the reservoir. If the reservoir is a human, then the portal of exit may be saliva, mucous membranes, feces, blood, or nose or throat discharges. By using barrier methods, such as condoms or masks, or covering the mouth while coughing, this link can be broken. The fourth link is the means of transmission. The pathogen can be transmitted either directly or indirectly. Direct transmission requires close association with the infected host, but not necessarily physical contact. Indirect transmission requires a vector, such as an animal or insect. The link can be broken through hand washing, safe sex practices, or avoiding contact with infected individuals. Link number 5 is the portal of entry. Entry of the pathogen can take place in one of 3 ways: penetration, inhalation, or ingestion. The level and severity of an infection may depend on the depth of penetration. Similar to the portal of exit, barrier methods, such as condoms or masks, can be used to break this link along with other methods, such as insect repellents. The final link is the new host. Once in the new host, various factors influence the severity of infection, including the strength of the immune system and the reproductive rate of the pathogen. Immunization, health promotion, and medical treatment can be used to break this link in the chain. An example of illness resulting from the chain of infection is the common cold. In this case, the pathogen is often referred to as rhinovirus. The reservoir is another person carrying this virus, who then propels the virus into the air via a portal of exit, such as a cough or sneeze. The route of transmission is direct to the new host, which takes place through inhalation, the portal of entry, of the virus. According to the Center for Disease Control and Prevention, knowledge of the portals of exit, entry, and modes of transmission provide a basis for determining appropriate control measures. In general, control measures are directed against the segment in the infection chain that is most susceptible to intervention, unless practical issues dictate otherwise. Potential interventions include controlling or eliminating the agent at the source of transmission, protecting portals of entry, and increasing the host's defense. An illness begins with the chain of infection. The first step in this chain is a pathogen, such as a virus or bacterium, that lives in a reservoir. The reservoir may be a human, an animal, or part of the environment. The pathogen exits its reservoir and is transmitted toward the new host. One method of transmission from a human reservoir is coughing. The pathogen then enters the new host. It may be inhaled or ingested. It may also penetrate through a mucous membrane or a wound. Disease control depends upon breaking one of the links in this chain. Understanding the pathogen, its portals of exit and entry, and its transmission is important. This understanding can help people identify the most susceptible link and focus prevention efforts on breaking it.
You are all excited about embarking on your new career in nursing. It is now finally your first day of working with patients. You are hoping that you remember everything you learned during your studies for becoming a nurse. Before worrying about what you need to remember about treating the patients, you need to remember how to be safe in a healthcare facility. After all, you won't be full of excitement and smiles if you catch a disease. This involves you remembering a specific set of regulations that are used to prevent the spread of infections and bloodborne diseases called standard precautions. You are very familiar with the precautions and recall that they are designed and implemented as a joint effort between two different government agencies. Let's see how this collaborative effort helps to ensure that you have a safe workday and patients have a safe visit to the healthcare facility where you now work. The first agency that has a vested interest in standard precautions is the CDC. This stands for the Center for Disease Control and Prevention. The CDC is the government agency that studies diseases and how to prevent the spread of diseases. It was the CDC that originally came up with a set of regulations known as standard precautions. These precautions were developed by the CDC based on research that showed that certain diseases are bloodborne and can therefore be passed between persons through exposure to blood and other bodily fluids that contain blood. Especially important in a healthcare setting because bodily fluids are handled regularly. The more bodily fluids are handled, the more the precautions became necessary in order to prevent the spread of infections from one patient to the next or from the patient to the healthcare worker. As a result, the CDC became very focused on making sure healthcare workers had knowledge of their role in disease prevention. The CDC's main role is to make sure nurses and other healthcare professionals know how diseases are spread and how to stop the spread. Each of the standard precautions are written and implemented with that goal in mind. The CDC is concerned with knowing how to keep everyone safe from diseases. On the other hand, we have OSHA, which is the Occupational Safety and Health Administration. You should notice that the word occupational is in the name of this agency. That is because they are concerned with people when they are working. This is an agency that provides regulations for employers to maintain safe working environments for employees. As you may have surmised at this point, OSHA is concerned with the safety and health of healthcare workers. OSHA originally came up with a set of regulations known as universal precautions that had pretty much the same information and precautions as standard precautions. Since the information was so identical, the precautions were merged and simply referred to as standard precautions. OSHA's goal in regards to standard precautions is to make sure places that employ healthcare workers, such as hospitals, doctor offices, labs, etc., provide the equipment and means for healthcare workers to follow each of the precautions outlined by the CDC. OSHA will visit the various healthcare facilities looking for items such as sharps containers, face masks, gowns, gloves, etc. They do announced and unannounced visits to check for compliance with the requirements to provide the necessary items to allow workers to protect themselves from exposure to blood and other bodily fluids. If a facility is not in compliance with the standard precautions, then OSHA can do anything from putting the facility on probation to fining the facility per violation to closing the facility down until they meet the requirements. So let's sum this up. A specific set of regulations that are used to prevent the spread of infections and bloodborne diseases is called standard precautions. The Center for Disease Control and Prevention, abbreviated as CDC, is a government agency that studies diseases and how to prevent the spread of diseases. The CDC's main role is to make sure nurses and other healthcare professionals know how diseases are spread and how to stop the spread. OSHA, the Occupational Safety and Health Administration, is the agency that provides regulations for employers to maintain safe working environments for employees. OSHA's goal in regards to standard precautions is to make sure places that employ healthcare workers provide the equipment and means for healthcare workers to follow each of the precautions outlined by the CDC.